Good morning, everyone. So in the next 20 minutes, I will try to walk you through the journey of first approvals of several complex generic long-acting injectable products. Hopefully, I will tell a good story. So at the end, you'll be able to describe different types of long-acting injectable products to recognize what are the regulatory and scientific challenges we have experienced, and some of them have been resolved, and we certainly recognize there are remaining hurdles, and have better understanding on what is the product-specific guidance and their role for supporting generic development. And lastly, but not the least, to be able to have a better sense of how the importance of GDUFA funded research program and how much that really contributed to where we are today. So long-acting injectable products, those are the products that are designed to provide controlled and sustained release for days and months. So in this slide and next couple of slides, I'm sharing information data that published in a review paper I co-authored with my colleagues, Dr. Wenlei Zhang, sitting in the audience, and two industrial peers back in 2021. We aimed at giving a very comprehensive overview of the landscape of long-acting injectables, including new drugs and generics in 2021. So the figure I'm showing here to show you the LAIs and the therapeutic areas that have been used. And you can tell they are very, very beneficial in the therapeutic areas that would require chronic disease treatment or management, because they can significantly improve patient compliance. Compared to conventional dosage forms such as tablets or capsules, we have far less numbers of applications approvals, but then the complexity level and the diversity of dosage forms is also very impressive. So as shown here, I'm gonna walk you through a little bit like history of the LAIs here. So back prior to 882, the very first generation or generations of LAIs could trace back as oil solutions and then some micronized drug substance suspensions, such as penicillin G. So back then, it was already probably very challenging to bring those drug substances into a product that can be given to the patient. But I don't think back then, now it's 40 plus years ago, the scientists then had really clear concept of being long-acting drugs. I think the very first, by definition, long-acting with a very defined therapeutic dosing regimen should be the polylactide co-glycolide PLGA-based microsphere product, Lupron Depot, which was approved in 1989. So followed on that in terms of dosage form, in 2009, there was very first a multivesicular liposome product. But that one was to deliver cytarabine, and then after that there was another MVL. But those two products, unfortunately, were very short-lived, not due to safety or efficacy, maybe more due to business reason. Then in 2009, going back to PLGA-based delivery system, the very first in situ forming gel or implant was brought to the market as Eligard to deliver the same peptide leuprolide acetate as the microsphere product in 2009, as shown on the top right panel. And that product is organic PLGA polymer solution, and the peptide is suspended in the organic given as an injectable suspension. Then upon administration, the dosage form would form a depot on the site. Then after that approval in 2011, the history always repeats itself, right? So it goes back to the drug substance suspension. And this time it was for paliperidone palmitate, which is antipsychotic, and it's the nano size instead of micro-sized suspension product. Follow that, MVL came back as what was mentioned in the commissioner's opening remark to market. This time is a very, very successful product for pain management, the bupivacaine multivesicular liposome. So in the 2021 paper, when we worked on it, we had a table to list all the high revenue LAI products. And back then, we still did not have any generic products approved. But that time we were already hopeful, because after that time was almost close to 10 years work, we already have in-house ANDAs that were under assessment. So we were very hopeful to see the first approval, which actually not one, we had three. We had three first generic approvals for LAI products for three separate ILDs. So those products were Sandostatin LAI, which is a peptide-containing microsphere product, and risperidone Costa, containing risperidone for antipsychotic treatment, and Vivitrol, which is an naltrexone microsphere product. So the Sandostatin LAI product is the product that containing a glucose PLGA polymer, which is very unique. Majority of the microsphere products on the market are using linear PLGA as shown here. So in the next couple of slides, I'm gonna share some of the regulatory challenges and scientific challenges we have had. So the regulatory challenge for PLGA-based LAI generics, because those are injectable products, parenteral. So per regulation, they need to be qualitatively Q1 and quantitatively Q2, the same as the reference listed drugs. However, for complex polymeric excipients in general, if you work with polymer, you know it's never a clean cut, right? The characteristic of the polymer is often a distribution, for instance, for the molecular weight. And the composition can also have certain variability. But then when it comes to PLGA, another layer of complexity is the products that are made by PLGA are really products by manufacturing. So depending on the manufacturing condition, methods, the raw material you start with may not represent the polymer you have in your finished product. And I'm having this figure here. It's in, I think, the Lupron Depot patent. By Alchemist, showing you, depending on also your commercial scale, the level of degradation PLGA can undergo is very different. So the regulation tells us, okay, you need to have the same inactive in the generic product, but it doesn't define what is considered as Q1 the same, and it's up to us to use the science to inform the decision making. And when we first started our research back in 2013, with the support of implementation of GDUFA, we knew nothing. That was the truth. So that regulatory challenge is translated into our scientific challenge. One, how do we define polymer sameness? What to do? PLGA was not new, and PLGA was very well known for formulation scientists. It started as a medical device for suture, and pioneered by, I think, Dr. the Lambert in MIT to be able to make high molecular PLGA that is suitable for drug delivery. But most of the time, people do it to develop formulations by error and trial and error. So there is really no very systematically methods available to characterize the PLGA and to inform. So we took the challenge and we funded our research program to look how we can reverse engineer the complex drug products and to identify the PLGA and determine its characteristics. And we published our results in a timely manner to facilitate generic development. Then I mentioned one of the first three approvals, the octreotide Sandostatin LAR, used a glucose core branched PLGA polymer. And there was zero literature published back then discussing how we can potentially characterize this particular polymer and to prove if the glucose is really in the core. And when we first started working on this project with Dr. Ian Park at Purdue, they just used the conventional NMR, and we didn't see any signal. So we were like, what's going on? And is it really a glucose core branched polymer? And how do we characterize the branch frequency? All those things were very challenging. And honestly, when we first started, we didn't know if we'd be able to deliver, but we have to start and just keep going. But at the end, we were able to actually publish multiple papers providing characterization methods to characterize the branch structure of the polymer. And we also actually characterized all the commercially available raw materials to guide the generic development. Then, so this slide just summarizes what we have learned and our current standard to guide the Q1 polymer sameness assessment. So our expectation is we would like to see the polymers extracted from the finished products, both T and R, to compare their properties to ensure they have comparable characteristics. This is not a conventional approach or a routine ask for all polymeric excipients. This is mostly, like I mentioned, we want the applicant to really keep the endpoint at the starting line to realize your game is not simple. Sometimes we see data submitted on raw material, but if you are really having a raw material matching on face value, the properties in the finished ILD product, when your material goes through manufacturing, you may not end up with a product that has comparable performance. So it's really for the applicant's benefit to know this in advance. The Q1 sameness, at least for most of our products, we think we have done great progress and have resolved most of the difficulties, if not all. But then in recent years, we start to see, because PLGA polymers, they have well-established clinical safety profile. So whenever a company can leverage from this group of polymers, they tend to stay within this group of polymer without trying new excipients. But then it's not sometimes you use one kind of polymer could give you the desired release characteristics. So then there are plenty of smart people, right? So what they do is they start to mix different PLGA polymers to fine-tune the degradation profiles to get what they want in terms of the release characteristics, which is brilliant. But then it's also going to be tremendously challenging for generic perspective. The first question we have is, if there's a mixture of two PLGA polymers, and how is it scientifically feasible for us to actually separate them? And if we can, to what extent? And what would be the most reasonable regulatory expectations in terms of the sameness in generics? So sometimes we do research, and that could, on face value, scare generic industry in terms of what are you trying to do. But sometimes we do things to the bottom because we want to know where should we draw the line. So we do something doesn't mean we're going to require the exact same thing as part of the generic development. But sometimes it's actually the opposite. If we have enough understanding, for instance, to show you don't really need to have the very detailed level of separation, and as long as you can meet the bulk property, that's sufficient, then that's our internal justification to develop the policy. So this is still ongoing, and we have made good progress, but we are not there yet in terms of clearly articulate where we stand when a mixture is used in a formulation. So next, the scientific challenge is the in vitro release testing. So this is a general challenge for long-acting products because the IVRT itself just very long, and because the release mechanisms also can be very complicated. So sometimes depending on the method condition, for the same formulation, you can get very, very different profiles. I think yesterday there was a good discussion in the IVRT session, right, talking about then what should we aim for? Should we aim for like a profile condition that mimic the in vivo PK? Or should we have a condition that we know that is more discriminatory for differences in formulation characteristics, composition, or manufacturing parameters? I think those are all very good and valid questions. And that's part of our research to really first to explore all the scientifically sound methods what are out there, and then what may be more suitable for QC versus for BE purpose. So here for long-acting injectable, we have been working on, for instance, looking into real time and then also accelerated time, because we understand how time is valuable for drug development. So we looked into develop important tools to understand the impact, how the formulation parameters, like the source of the polymer, if you purchase from supplier A versus B, if they give you the same performance, and then also use that to explore novel B approach. For PLGA-based microsphere products, we're not there yet, but we have ongoing research for instance, in situ forming, which we are trying to see if we have enough understanding to push for totality of evidence-based in vitro only approach for supporting BE, or as a in between, if we can have in vitro in combination with in vivo, then reduce some level of burden at the clinical site. And ideally, once we have methods, IVRT methods that we're really comfortable, we will start to maybe implement them into our PSG to be more descriptive so company can follow. And we also start to include references in our PSG so company can directly look at the papers we have published. So challenge 3, bioequivalence study, is already sort of part of the IVRT. In general, the BE study, our expectation, it should be accurate, sensitive, and reproducible approach. So for PLGA-based long-acting drugs, so far, most of the PSGs we recommend in vivo BE studies with PK endpoint. We have one PSG for a dental product that is a locally acting product to recommend comparative clinical endpoint study. And we actually understand and know the challenges associated with that approach. But that was the most viable approach at the time of PSG development. And I think that goes back to another question raised in yesterday's session, why FDA cannot just recommend a PSG with the most sound approach instead of keep revising it. But I think the fact is, when something is complicated, it's complicated for everybody, for industry and for FDA. So it does take everyone's time to really understand and come up with an approach and keep improving upon it. And that was what we have been doing. So for the dental powder product, we have been using that as a model product to study, and we're really getting close to include an alternative in vitro approach. So this slide here just give you an example PSG. This is with partial AOC, because for long-acting products, we often also recommend partial AOC instead of just conventional PK, excuse me, to make sure the PK profiles are comparable, but also the sustainability of a product is also similar. Sorry, excuse me. Then the next scientific challenge or efforts, I would say, we have been working on is the IVIVC. So here I mentioned a couple of times already, totality of evidence in vitro approach. So IVRT is always part of the approach, but because that is a totality of evidence approach, so as far till today, the PSGs for complex LAI products, in which case we recommend in vitro only approach, the IVRT was not targeting IVIVC. So IVIVC is always desired, and it in theory can be a standalone study. But as totality of evidence approach, the IVRT is more targeting to demonstrate understanding of the drug release mechanism instead of really strictly targeting IVIVC conventionally to predict the PK profile. So there is a subtle difference. I'm not discouraging IVIVC, and it can be very beneficial, but just want to point out our totality of evidence, the IVRT, is not necessarily say you have to have an IV-IVC. But in order to develop IV-IVC, so the paper I'm showing here was published by Dr. Diane Burgess in UConn, and they have lots of experiences exploring IV-IVC for complex LAIs. And in recent years, we also start to collaborate and exploring advanced analytical technology to better understand the microstructure of those polymeric systems, so that can inform really the release mechanism. So I'm showing you a few figures here. So we use, for instance, the iron beam focused SEM, and then we take the image and use artificial intelligence informed data segregation to reconstruct the inner structure, and then couple that with our IVRT data to really understand the drug release mechanism. So this slide here, I just give you a quick walkthrough in terms of the timeline of our GDUFA research efforts and how OGD FDA progressed to where we are for the first generic approval. So back in 2013, with the implementation of GDUFA 1, we initiated our first batch of research activities focusing on PLGA-based LAI products. Then in 2015, the first paper published describing analytical methods that can be used to reverse engineering the PLGA-based RLDs. And then in the same year, we revised previously published PSG for risperidone Costa to include specific recommendation on IVRT and further updated our recommendation for the PK study portion. And then the first guidance for octreotide LAR, naltrexone and risperidone was published or further revised in LDs. Besides the ENDA assessment and our PSGs, then the research outcomes has also been used to address Yeah, I guess the milestone slide wants to have a little bit more highlights. Okay, so the next couple of slides, really I just quickly mention the MVL, which you have heard multiple times. So this is the picture again, and this is a mixture of liposome and microspheres. So the particles themselves are more like microparticles, and they are really in the micron range, and they are sphericals. But then within the spherical, you have this honeycomb structure where the lipids are forming small compartments or chambers. And then that particular packaging has the conventional liposomal feature. So the complexity of this bupivacaine MVL is a lipid-based microparticle with nano-sized inner structure. It's very complex in terms of manufacturing. I will show you, we started our internal research, I think back in maybe 2015. And the FDA lab, we found maybe close or more than 30 ways to fail the manufacturing. So that's how many times we have tried and failed. And it's really a big shout out to the lab fellows. Just don't give up until we had a very decent formulation that has this honeycomb structure with reasonable stability. And then this is a locally acting product. Systemic PK is also surgical site dependent. Yeah, then in 2017, we published a paper presenting a very novel IVRT method. Until today, we haven't seen a replicate. What's unique about this method, which made the cover story of Journal of Control Release, back then is it's a testing method coupled with inline UV measurement to continuously monitor the drug release. And we were able to find a condition that really reflects the multi-physic release profile of this MVL product. Its PK profile, as you can read in the labeling, is multi. It has 2 peaks. So it does have a lag phase before the secondary peak or drug is released. Then based on our research, we published our PSG in 2018, and then the first generic was approved earlier this year. But then you can tell from 2018, we didn't really stop there. We know what we recommended in our PSG, and back then we didn't really have all the data to prove the feasibility, or also we want to help generic to better understand our methods. So we had external research carried out reverse engineering and characterization, and we published a paper sharing all those analytical methods. So in today's presentation at the end, I don't have an acknowledgement slide because it's really just not possible for me to have one slide listing all the names, all the individuals that have contributed to the LAI research area and internally and externally. So as a tribute, I'm having 2 slides listing over 40 publications that were produced through the GDUFA research by our internal external collaborators, and also can be a good source for the audience if you are interested in reading some of the papers and learning more about what we have learned. And for my challenge question, it's very simple. Just want to see if everyone thinks all long-acting drug products are considered complex products. True or false? Yeah, it's false. Because like I mentioned, the very first generations, the oil solution, those are not complex. Then for drug substance suspensions, they are complex products and they are very challenging to develop as new drugs. But within that, depending on the clinical risk, penicillin G versus paliperidone, we will not consider them at the same complexity level. So what I want to say here is really, when we think something is complex or not, you really have to take this holistic, 360 angle to look at all the aspects rather than fragment and just look at one aspect. So with that, I will summarize. Polymer or lipid-based microparticles, those are very complex, long-acting injectable products. GDUFA research projects have made great contribution to improve the understanding of these products. Those outcomes we have used to develop our product-specific guidances and to address the regulatory inquiries to support the generic development and approval. And OGD will continue to improve PSGs based on updated understanding obtained through communications with generic industry, workshops, CCs, pre-and meeting requests, and GDUFA-funded research. So I think this is really a collaborative effort. We take the first step to publish a PSG to reflect what we think at the moment maybe the best approach. But that doesn't mean that is truly the best per your perspective. So we really need to hear industry's feedback, criticism, challenges. Those are fine because those are the things push us forward, and we should work together and communicate more. So that's why we highly encourage generic industry to really leverage our GDUFA research outcomes and engage with us early during development. So with that, I would like to thank you for your attention and invite all the speakers to the podium. Thank you.